Hi students, today we will learn about the physiological anatomy and neuronal activity of the cerebellum. Coming to physiological anatomy of cerebellum, externally has superior surface, inferior surface and folia and primary fissure, horizontal fissure and posterior lateral fissures. So you can see this is the primary fissure. This is the primary fissure. This is the horizontal fissure. Okay. So primary fissure here this is the horizontal fissure. So these are the two hemispheres of the cerebellum. Here you can see this is from the superior side, this is from the inferior side surface. So you can see on the superior surface vermis, anterior lobe, primary fissure, posterior lobe and horizontal fissure. Inferiorly you can see flocculonodular lobe, posterolateral fissure, floccules and nodules, vermis and posterior lobe. Coming to anatomical parts of the cerebellum. So, coming to lobes of the cerebellum, anterior lobe, posterior lobe. So, anterior lobe contains lingula, central lobule and culmen. Okay. So, here you can see lingula, central lobule and culmen. It is composed of anterior lobe. Then primary fissure. Then this is posterior lobe. So posterior lobe contains declive and folium. Then comes horizontal fissure. Then it comes tuber, pyramis, uvula and nodule. Okay. Here anterior lobe, for the anterior lobe there is no lateral projection for the lingula. Okay. And central lobe will be projected to ala and culmen is projected to anterior quadrangular lobule. Here declive is projected to posterior quadrangular lobule and folium is projected to superior semilunar lobule. And after the horizontal fissure, tuber is projected to inferior semilunar lobule and pyramis is projected to biventral lobule and uvula is connected to tonsil and flocculus. Here this is the flocculus, it is connected to nodule. Okay, so these are the parts of the anterior lobe and posterior lobe. Then coming to anatomical divisions. So this is vermis and this is the hemisphere. Okay, this is the anterior lobe we have already seen and this is the posterior lobe and this is the flocculonodular lobe. So anatomically it is divided into three parts: anterior lobe, posterior lobe, flocculonodular lobe. Phylogenetical divisions. Phylogenetically, it is again divided into three lobes. That is, flocculonodular lobe is nothing but archicerebellum. Anterior lobe is nothing but paliocerebellum. And posterior lobe is called as neocerebellum. Okay, archicerebellum, paliocerebellum, and neocerebellum. So, posterior lobe consists of neocerebellum, middle divisions of the vermis, and their lateral extensions. This is anterior lobe. This is total thing is posterior lobe. This is flocculonodular lobe. So flocculonodular lobe is called archicerebellum. This posterior lobe is called neocerebellum. The vermis and is the lateral extension. and anterior lobe called paleo-cerebellum. Functional divisions. Functionally, it is again divided into three lobes. That is spinocerebellum. That is vermis and intermediate zone of the hemisphere. That is spinocerebellum, and this is vestibulocerebellum. That is flocculonodular lobe. Okay? Flocculonodular lobe. That is called vestibulocerebellum. And all this spinocerebellum, you can see spinocerebellum, you can see vestibulocerebellum, okay, that is flocculonodular lobe, and this is cerebrocerebellum. So functionally it is divided into vestibulocerebellum, spinocerebellum, and cerebrocerebellum. Cerebrocerebellum is composed of neocerebellum, that is posterior lobe. So you can again see here functional division is written here. This vestibulocerebellum is nothing but cerebellum. It is composed of flocculonodular lobe anatomy and immediately adjacent parts of the vermis. So what is the role of the vestibulocerebellum? The vestibulocerebellum regulates the balance and eye movements. It receives vestibular input from both the semicircular canals and from the vestibular nuclei and sends its fibers back to the medial and lateral vestibular nuclei. Then it also receives visual input from the superior colliculi and from the visual cortex. Regions of the vestibular cerebellum cause disturbances of balance and gait. Okay? So, it is helpful for balance and eye movements and gait. Okay? So, it receives input from the semicircular canals and vestibular nuclei and superior colliculi. Then again, it sends its information to the medial and lateral vestibular nuclei. This for balance and eye movement. Then coming to spinocerebellum is also called paleocerebellum. Vermis and intermediate parts of the hemisphere, that is paravermis. Vermis and paravermis are included with the spinocerebellum. It's nothing but paleocerebellum. Why it is important? The spinocerebellum regulates body movements and limb movements. It receives proprioceptive impulses from the dorsal column, okay, through spinocerebellar tract and also from the trigeminal nerve as well as from visual and auditory inputs. It sends fibers to the deep cerebellar nuclei, that is to the fastigial nucleus, and it projects to cerebral cortex via the midbrain thalamus and to the brainstem via the reticular formation in the pons and vestibular nuclei in the medulla oblongata, thus providing modulation of the descending motor systems. Okay? This spinocerebellum contains sensory maps as it receives data on the position of various body parts in space. In particular, the vermis receives fibers from the trunk and proximal portions of the limbs, while the intermediate parts of the hemisphere receives fibers from the distal portions of the limbs. The spinocerebellum is able to elaborate the proprioceptive input in order to anticipate the future position of the body, okay, during a movement in a feed forward manner. So it is helpful for regulating the body movement and the limb movement. Then coming to cerebrocerebellum is nothing but neocerebellum. the lateral parts of the hemisphere are included in this. The neocerebellum is involved in the planning of the movement and it evaluates sensory information for its action. It receives input exclusively from the cerebral cortex, especially from the parietal lobe via the pontine nucleus, okay, and dentate nucleus in the cerebellum, and sends fibers mainly to the thalamus, that is ventrolateral thalamus, and to the red nucleus. Okay. The neocerebellum is involved in planning the movement is about to occur and has purely cognitive functions as well. So it is involved in planning the movement and it evaluates the sensory information also. Coming to histological structure of the cerebellum, you can see this is the cerebellar cortex. It is folded many times. These foldings are called folia. There are 5 main types of neurons in the 3 layers. So cerebellar cortex has 5 main types of neurons in 3 layers. What are those 3 layers? Cerebellar cortex has 3 layers, has 5 main types of neurons. And you can see after cerebellar cortex. First cerebellar cortex. What are those 3 layers? Molecular layer, granular layer and Purkinje layer. The molecular layer, Purkinje layer and granular layer. There are 3 layers. And after this cerebellar cortex, we have white matter, and in the white matter, there will be deep cerebellar nucleus. Coming to cerebellar cortex, you see cerebellar cortex, the gray matter of the cerebellar cortex has molecular layer, Purkinje layer and granular layer. What are those 5 types of cells? The molecular layer, you have stellate cells and basket cells. The Purkinje cell layer, you have Purkinje cells. The granule layer, you have granule cells and Golgi cells. This is the white matter which contains deep cerebellar nuclei. It has climbing fibers which arises from the inferior olivary nucleus and mossy fibers which arises from the other nucleus. So you can see here stellate cell is like this and basket cell is like this. There are many other types of cells also. These are the main cells. Molecular layer is composed of 2 types of neurons or cells, stellate and basket cells, and it has unmyelinated nerve fibers. The stellate cells are star-shaped and they are more superficially located. Their dendrites synapse with the parallel fibers of granule cells and their axons synapse with the dendrites of Purkinje cells. Next, coming to basket cells. Basket cells are located deep in the molecular layer. They receive input from the parallel fibers. Their axons branch and form a basket around the cell bodies of Purkinje cells, hence the name comes. Each basket cell will synapse with 70 Purkinje cells. Nerve fibers present in the molecular layer are parallel fibers. These parallel fibers are the axons of granule cells and dendrites of Purkinje cells and climbing fibers from inferior olivary nucleus. All these are the nerve fibers present in the molecular layer. You see this Purkinje cell, how it looks like. The dendrites of Purkinje cells extends into the molecular layer and provide axodendritic synapses. Okay. Then axons of these cells make synaptic connection with the deep cerebellar nuclei in the medullary bone. They act as the sole output neurons from the cerebellar cortex and they are inhibitory in nature. Okay, they will exert inhibitory influence on the deep cerebellar nuclei. Next comes granule cell and Golgi cell, how they are looking like. You can see The granule cell layer consists of granule cells and Golgi cells with their processes and sensory mossy fibers with their synaptic glomeruli. The granule cells are very small and numerous, okay, and has numerous spherical neurons. Axons of the granule cells ascend into the molecular layer and form the parallel fibers that will make excitatory synapses with the dendrites of Purkinje cells, stellate cells, Basset and Golgi cells. So only these cells are excitatory. Now coming to Golgi cells. Golgi cells are large cells and are less numerous. The dendrites project to the molecular layer and receive inputs from the parallel fiber. Their cell body receives inputs via collators which are coming from the climbing fibers and Purkinje cells. Their axons will branch extensively and make inhibitory synaptic connection with the dendrites of granule cells. So this is the diagrammatic representation of the synaptic connection, the cerebellar cortex. You can see here in the molecular layer we have stellate cells, okay, and basket cells. So these basket cells will form a basket around the Purkinje cell. Here comes the Purkinje cells. Then comes the granular layer which has granule cells and Golgi cells. This granule cell, it makes excitatory synapses. Okay, excitatory synapses with the dendrites of the Purkinje cells, and they will form parallel fibers. So parallel fibers are formed from axons of granule cells. Then comes Golgi cells. So Golgi cells are larger cells and they will be inhibitory in nature. The dendrites of granule cells. Next comes sensory inputs to the cerebellar cortex. So there are two sensory inputs. One from the inferior olivary nucleus. They are climbing fibers. So they will climb up to the topmost layer, molecular layer. Then comes other nuclei, other cerebellar nuclei and other nuclei. These are mossy fibers. Okay. This is a climbing fiber. This is mossy fiber. Mossy fibers will synapse with the granule cells. Next coming to simple spike and complex spike. So this is a simple spike. This is complex spike. So simple spike is formed from parallel fiber input. Okay. Simple spike is formed from parallel fiber input. And this complex spike is formed from climbing fiber input. Okay. Cerebellar Purkinje neurons communicate downstream circuit elements by generating two distinct types of electrical activity. Okay. So this is the communication of the Purkinje neuron downstream. The Purkinje neurons fight conventional action potentials. They are called simple spikes. Okay. And they also intermittently fire a highly stereotyped spikes, they are called complex spikes. Okay, so when they are activated by climbing fiber input, then they will give action potential like a complex spike. Simple spikes occur at a very high frequencies in the range of 50 spikes per second and are driven by pacemaking ion channels expressed by Purkinje neurons. The high simple spike rate is then modulated by excitatory and inhibitory synaptic input, whereas the complex spikes occur in response to excitatory synaptic input from the climbing fiber. These compound electrical events are driven by large voltage gated calcium conductance in the dendrites of Purkinje neurons. So this is because of opening of voltage gated calcium channels in the Purkinje neurons. Finally, the two forms of excitabilities will interact. Complex spikes can exert indirect effects on simple spiking rate. Together, these two firing behaviors endow the Purkinje neurons with a range of signaling behaviors critical to cerebellar contributions to motor coordination and motor learning. So, they are important for motor coordination and motor learning. The climbing fibers arises from inferior olivary nucleus, which is situated in the medulla and reach the cerebellum via olivocerebellar tract. And the climbing fibers will establish one-to-one connection with the Purkinje cell dendrites and they will excite the Purkinje cell. The collaterals of climbing fibers synapse with all other types of neurons in the cerebellar cortex and also with the deep cerebellar nuclei. Whereas the mossy fibers, unlike the climbing fibers, the mossy fibers have many sources of origin. For example, axons of the spinocerebellar pathway, vestibulocerebellar, reticulocerebellar, uniocerebellar, and corticopontocerebellar tracts. Mossy fibers, why they are labeled like that? Because they will resemble like a moss plant. Each mossy fiber makes synaptic connections with the dendrites of many granule cells, forming synaptic glomeruli. So they will form synaptic glomeruli. The glomeruli also contains inhibitory ending of Golgi cell. Each mossy fiber activates about 450 Purkinje cells through the granule cells and their parallel fibers. Thus, a climbing fiber excites a single Purkinje cell, whereas a mossy fiber through the granule cell it will excite several thousand Purkinje cells. Both climbing and mossy fibers of sensory inputs are excitatory in nature. Out of the 5 types of neurons we have seen in the cerebellar cortex, only granule cell is excited. It releases glutamate as a neurotransmitter. The other 4 types are inhibitory and they will release GABA, that is GABA aminobutyric acid neurotransmitter. Then coming to white matter of the cerebellum, there are projection fibers, those forming inferior, middle and superior cerebellar peduncles and association fibers and commissural fibers. Then coming to deep cerebellar nuclei, you have 4 deep cerebellar nuclei. This is the dentate nucleus, this is the emboliform nucleus, this is the globose nucleus. Both together, globose and emboliform together called nucleus interpositus, and this is called most medially fastigial nucleus. Then coming to neural circuits and neuronal activity of the cerebellum. So you have already learned that afferent fibers to the cerebellar cortex coming from climbing fibers and mossy fibers. Then here you should remember that there is 2 feed forward inhibitions and 1 feedback inhibition. This is feedback inhibition via the granule cell. Okay? For the granule cell feedback inhibition. This is the feed forward inhibition to the Purkinje cell and granule cell. Okay? So in feedback inhibition, what is happening? The neuron which is gets activated will inhibit the neuron which is activating that. So that is the granule cell. For example, it is the granule cell. And feed forward inhibition, one neuron will activate both this and this, but this blue neuron will inhibit the gray neuron. So this is a feed forward inhibition. It is present for granule cells and Purkinje cells. Here you can see feedback inhibition of granule cell. So and feed forward inhibition of granule cell and feed forward inhibition of Purkinje cells. So mossy fibers will excite the granule cell. Mossy fibers excite the granule cell. Then this will excite the Purkinje cells. But this will also excite the basket cells. In turn, these basket cells will the Purkinje cells. This is the feed forward inhibition of the Purkinje cell. Then coming to feed forward inhibition of granule cell. So what is there in it? The mossy fibers will excite the granule cell, and these mossy fibers will also excite the Golgi cell. This Golgi cell will inhibit the granule cell. Okay. This is the feed forward inhibition of granule cell. The third one is feedback inhibition of the granule cell. So granule cell will inhibit the Golgi cell, and Golgi cell will inhibit the granule cell. Reverberating circuit, a circuit of head. This is the reverberating circuit of the head. So it is helpful for rapidly occurring supination pronation movements. Okay. So it is shown here. So from the inferior olivary nucleus, climbing fibers will be arising. Okay. They will excite the Purkinje cell, and from other sources, mossy fibers will be coming and they will excite the granule cell, and they will form the parallel fibers and they will excite the Purkinje cell. Then the Purkinje cell inhibits the deep cerebellar nuclei. So this is the normal phenomenon. So what happens when if a person is doing rapidly pronation and supination? So all these things will be occurring, but one this whole system will form an echo of the other system. So this is called a reverberating circuit of head. Next, coming to neuronal activity of deep cerebellar nuclei. The deep cerebellar nuclei are excitatory have excitatory neurons. These nuclei receives inputs via collators from the climbing fibers, mossy fibers and other inputs. These sensory inputs maintain the DCN, that is deep cerebellar nuclei, always in a excitatory state. In turn, these nuclei sends excitatory impulses to thalamus and brainstem nuclei. So, however, this Purkinje cell output is inhibited into the DCN via GABA. So, the Purkinje cell inhibits the activity of the deep cerebellar nuclei and vestibular nuclei also. This is the afferent and efferent connections of the cerebellum through peduncle. So, we have three peduncles: superior, inferior and middle. So, from the superior cerebellar peduncle, what will come? Anterior spinocerebellar tract will come. Okay. So, it will carry information from the lower extremity of the cerebellar cortex and nuclei. It is ipsilateral. Okay. Cell bodies are in the Clax column. From the middle, you have pontocerebellar tract. Okay. So, here it is crossing to the opposite side. Okay. Location of the cell body is in the pons. From the pons to cerebellum, pontocerebellar tract will go through middle. So only afferent will be there, the Middle cerebellar peduncle. No efferent. Here, in the inferior cerebellar peduncle, you have olivocerebellar tract, vestibulocerebellar, posterior spinocerebellar, cuneo-cerebellar, rostral spinocerebellar, reticulocerebellar, reticulocerebellar, trigeminocerebellar tracts. And coming to the efferents from the cerebellum, from the inferior cerebellar peduncle, we have cerebellovestibular tract to the vestibular nucleus. And from the superior, we have cerebellorubral, cerebellothalamic, uncinate fasciculus, and nucleolivary tract. They are going all to the concerned positions. So the next video is about functions of cerebellum.